I'm very happy to welcome everybody to the third webinar of the online edition of the Protein Electrostatic Conference. I'm extremely happy to say that the number of registration is increasing very steeply. So we have at the moment more than 140 registration plus 20 subscription to the YouTube channel. I am happy to see that I'm asking you to subscribe even many more, because if we overcome the 100 of subscription, we can definitely assign a name to the channel, and we could be able to use this channel to share as a resource, to share information and disseminate messages in the community even after the series of webinar has finished. So this could be a nice thing. So I remind you, but if you are here, you already know that it is possible to attend actively on Zoom or to watch on YouTube. I'm sorry, I have to change sometimes the YouTube link because of a mismatch between Zoom and the YouTube channel. I hope we will fix eventually this issue. So I had also today to change the YouTube link on the website. Everything should be smooth now. We had a feedback form on our website. So if you have comments that you want to leave them, it is possible to do there. And I will again encourage, strongly encourage the youngest colleague to submit the abstracts for a flash talk. So I would like eventually to have one long talk and one short talk per presentation per day. And so I told you last time that we had to fix some organizational issues of overlaps that may lead to a change of day. Actually, we found that it would not be necessary to change the day, but we will delay the starting of the webinars of just 15 minutes. So from the next week, we will be starting a quarter of hour after the starting that is now. So it's not going to be a big change. Everything will be updated on the program on the website. Okay, so let me introduce the speaker of today, who is Professor Stephen Boxer. He is the Camille Dreyfus Professor in the Department of Chemistry at Stanford University. He is an elected fellow of the American Academy of Arts and Sciences, the Biophysical Society, and the National Academy of Sciences. His research interests are in biophysics, at the interface of physical chemistry, biology, and engineering, and they include electrostatics and dynamics in proteins, especially related to enzyme catalysis. So I am very happy to have him here and to give him the podium, virtual podium, so that he can talk about electrostatics and enzyme catalysis.
Oh, great. Thank you. Can everybody hear me? Is it okay?
Yeah. Yeah.
Okay. As I was mentioning at the beginning, we've got some problem with different screen sizes, so I'm going to stick with this because it works. Good morning to everybody. Good evening, wherever you are. And thanks to Walter for organizing this. I had really looked forward to meeting some friends, some of whom I see here, many of whom I don't know. In Genoa, but maybe sometime in the future somewhere. So I've just, in general, I've worked in the general area of electrostatics for a long time. We started looking at, pKs of buried charges and the effects of buried charges on redox potential way back in the 1980s when Marilyn was still in elementary school. No, actually, she was a graduate student, and I knew her when she was a graduate student. And we moved away from that, and I'll try to make clear why we moved away from it, because we moved towards a different way of describing what's going on and tried to introduce the language of electric field. And this will be very familiar to Lauren, who was a postdoc in my lab, who's heard this talk probably 8 times. So I'm going to try to, since it's a different audience, and since I think the emphasis has been a little different, I'm going to try to sort of step through this very, very systematically. Hopefully not bore you by doing that, but I want to make it really clear what we're doing and why we're doing it. So general background, then I'll specifically talk about the vibrational Stark effect, the Stark tuning rate. I'll even get into unit analysis and things of that sort, vibrational solvatochromism, how we get to absolute electric fields, and why that's important. And then I'll really focus on the electrostatic contribution to catalysis, which is one of the, I think, most important aspects of electrostatics. I'll focus on an enzyme we've worked on for quite a while, up until about late last year, when we published the last paper, probably, or I guess we have one more coming, ketosteroid isomerase. It's an incredibly simple problem, a very fortunate choice for many reasons. And I don't think I'll have the time probably, but I can just maybe just with one or two slides summarize where we are. So the next time we all meet, you'll know what I'll be talking about. We're working on sort of the evolution of electric fields in 2 dimensions, and aspects of excited state catalysis using GFP. So by way of background, I just put up this slide just for anyone who isn't familiar with this. This is a famous representation of the extraordinary efficiency of enzymatic catalysts done by a leader in the field, Wolfenden, a long time ago, showing that we have these unbelievable rate accelerations. So we're not talking about a factor of 2 or a factor of 5. We're talking about a factor of 10 to the 15th, even as much as 10 to the 25th rate accelerations. And one always has to reference rate accelerations to the uncatalyzed reaction, which is often hard to measure because it's so slow, but the uncatalyzed reaction in water. And of course, we teach in introductory chemistry courses, we teach that reactants go on to products through a transition state, and that what the catalyst is doing is somehow lowering the activation free energy. So this is all very familiar. What is often not really discussed in great depth is what is really responsible for lowering the activation free energy. And of course, this is a very general question in chemistry and biology. I mean, if you look at, let's say, industrial chemistry, as I've moved into this field, I've been invited to meetings with people who do heterogeneous catalysis, and heterogeneous catalysts, are responsible for roughly 80% of all chemical reactions that are currently used in manufacturing. It's really quite amazing. And almost in no case does one really know very much or understand very much about the true mechanism. So here really is really the most important slide, because it's really going to be the perspective we're going to take on connecting electrostatics, or electric fields would be the language I prefer, and catalysis. So again, I have a simple made-up reaction coordinate going from reactants through transition state to products. And I've made this up and this is completely made up just for simplicity, in which the reactant, since we're focusing on fields, we're going to be interested in dipoles and charges, polarizable entities. What is the reactant dipole? I'm just going to make it 0. Okay, I'm really talking about the dipole moment associated with the bond associated in catalysis, and I'll say more about that later. If we go to the transition state, there's often charge separation, as some of you like Marilyn know well, and I've worked on electron transfer a long time. That's a perfect example where obviously charge moves over a long distance. Proton transfer, it's the same thing, but this is true for almost all catalyzed reactions. So there's some dipole, and I'm using this simple dipole approximation. There are many approximations that are involved here, but I think they're all sort of physically reasonable. And we'll call that the transition state dipole. So the dipole moment difference associated with going from the reactant to the transition state we'll call the reaction difference dipole, delta mu of reaction. And that's just the transition state dipole minus the reaction dipole, which in this case is just the transition state dipole, because I defined this as being equal to 0 arbitrarily. So we're going to focus on this movement of charge, okay? We're not going to talk about how we get to the transition state. That's a very important separate problem, but that's not going to be the focus today. So the perspective that we're taking is sort of thinking about electric fields or electrostatic effects, is what is the field that this reaction sits in? Okay, and I'm just going to again simplify the field by using a vector representation. I'll just call it field with a vector. And it, of course, interacts with this difference dipole going from reactants to transition state. And I'm using, by the way, the physics convention here for dipoles. So everything is, the signs are all correct, not the chemist's convention. And if, as in this picture, the field were aligned with the reaction difference dipole, it would lower the transition state energy. But of course, it doesn't have to be that way. It's a dot product. It could raise it. It could be perpendicular. I've just, by way of illustration, made it this way. So this is the total field from the protein. It's everything that the reaction sort of experiences inside a catalyst and an enzyme in particular. Now, to put numbers on this, if we were to separate a charge, let's say by 1 angstrom, just again, a made up number, but not untypical of many reactions, and we put that charge or that dipole in a field of 10 megavolts per centimeter. I'll use those units. Physicists might prefer to use volts per meter, but spectroscopists use megavolts per centimeter. This may seem like a very large field. It's certainly a larger field than you can apply in a parallel plate capacitor without dielectric breakdown. But as we'll see, it's very much a small field compared to the fields that are present inside organized matter. But just again, using round numbers, 1 angstrom dipole and a 10 megavolt aligned with a 10 megavolt per centimeter field would lower the activation free energy by 10 kilojoules per mole. That's not a very big number, but of course it goes in the exponent, so it could have a substantial effect. And you see that it's linear in both of these terms. So if there was a larger reaction movement of charge or a larger field, both of those could make this effect substantially larger. And we'll see that the fields can be much larger, in fact. And so this effect can be very large. Those who come from a sort of chemical physics background may be familiar with this concept because it was discussed a lot back 30, 40 years ago. It was called the matrix electric field, and then just sort of disappeared largely from the literature, but it's really what the reaction feels. Now, of course, the question is, I mean, this is all fine and good. I can make up the story like this. The question is, what is the actual electrostatic contribution to catalysis, and is this really relevant? So this idea has been promoted a lot by theorists, in particular by Ariel Warshall. I'll show Ariel's words in a second, but experimentalists largely ignore this concept. And in fact, you'll rarely see this discussed, for example, in biochemistry textbooks, which always have a big chapter on catalysis. What you'll see is sort of vague references to electrostatics. And what we're trying to get away from is being vague about electrostatics. We want to be quantitative. So here on the left is an example of the activation energy. Now I'm in kilocalories per mole for an uncatalyzed reaction, which happens to be the one I'll talk about later. So roughly 22 kilocalories per mole. And in the enzyme, it's roughly 11.5 kilocalories per mole. So this is an example of lowering the activation free energy. And the question is, what's responsible for that? And there's a long history in this area, and it's people fight quite violently about this, as I've discovered moving into this field as a novice. Bill Jenks, who was a great mechanistic enzymologist, and really promoted the perspective that what's really involved here is bringing things together and positioning the components. He talked about the effective molarity of that association. To me, this sounds like an entropic contribution to catalysis. And you'll notice the dates here. These are really old ideas. If you look in a biochemistry textbook, largely what you'll see is this description of how enzymes work. On the other hand, and he was, of course, an experimentalist. On the other hand, if you look at people who come from a theory background, in particular Aria's work, and I think the words here are important because they really are what I've been talking about. Enzymes, on the other hand, can stabilize varied constellation of charges by fixing their dipoles in appropriate orientations. That's really what I was just describing. And then the final sentence here, the effect of charge stabilization is shown to account for most of the difference between the activation energy of the enzymatic reaction and the reaction of the corresponding model compound in water. So what Arya is describing in this paragraph, but you'll have a hard time finding it in his papers because his papers are complicated and difficult to read sometimes, is what I was talking about here with this simple picture. I'm oversimplifying Arya's work. Now, the real issue here is that, you know, first of all, he says most. I mean, those of you who know Arya, he's quite adamant about whatever he is thinking. And if you go to a conference, and I've been at meetings with Arya, you know, the descendants of Bill Jenks will say, well, no, this is unimportant or of minor importance. This is what matters. And the two sides are clashing. So, you know, our contribution to this field really is to come in and try to parse these effects and in a quantitative and, if possible, general way. But what's required then is, if we go back to this picture, is we can sort of estimate what the reaction difference dipole is just by looking at the mechanism and doing, you know, simple calculations of the sorts that organic chemists do all the time. But it's really this field that's the hard quantity. So how do we get to fields inside enzymes? So these are my hands. This is my only contribution to this work. Really, how do we measure fields inside enzymes? You're all familiar with these patriotic red, white, and blue electrostatic maps of proteins, but you know, and they're very useful. But the question is, what do they mean quantitatively? Now, spectroscopy in an electric field, of course, is called Stark spectroscopy. It's been around since 1913. And the Stark, and since this is a meeting in Italy, I think we should call it the Stark-Lasorda effect, because it was actually simultaneously done by an Italian scientist, Lasorda, who doesn't get credit for the Stark-Lasorda effect. And you all learned about this early on as the splitting of lines by an electric field. And we've been using the Stark effect mostly for electronic transitions for a long time. Now we're going to turn it around. Okay, we're going to actually use Stark spectroscopy to calibrate probes and then use those probes to actually tell us something about what the field is. Okay, so it's really the inverse. Okay, so that's all by way of background. Now let me get specific. And as I say, it's going to take me a while to get to the actual subject because I need to build up the sort of the infrastructure of how we are pursuing this problem. So first, the vibrational Stark effect. So the vibrational Stark effect, when we first became interested in this, and why we became interested in it isn't really relevant, we were told you couldn't measure it because we all, or this is a group of people I think whom probably all have certainly taken, but probably also teach quantum mechanics. And of course, we teach the harmonic oscillator. And for a harmonic oscillator, if you look, you'll remember that the average position of the oscillator is the same in all of the vibrational levels. And so if you go from one vibrational level to another, there should be no charge displacement. That's what gives rise to the Stark effect for any transition. But real potentials are, of course, anharmonic. And to the extent they are, there is a difference in dipole between the lowest vibrational and the second and the first excited vibrational level. So that's another delta mu, okay? But I don't want to confuse it with the delta mu of reaction. This is a spectroscopic delta mu. Now, we've developed methods, I'm not going to go into the experiments at all, to measure what delta mu is for a vibrational transition. We started this a long time ago, initially mostly looking at nitriles. Suffice to say, we put our sample into basically into a parallel plate capacitor. It's sort of an external electric field. This is the only part of this discussion that involves an external field. Almost everything else will be fields internal to the sample. And its interaction with this difference dipole, which could be an electronic transition, or in this case, a vibrational transition, leads to an energy shift. And through an analysis of these spectra, we can get out what the magnitude is of delta mu for this vibrational transition. And the typical unit for dipoles is dBi. A more useful unit, and this will be clear in a minute why this is useful, is the Stark tuning rate. And these are isodimensional. And the Stark tuning rate has this sort of weird units of wave numbers per megavolt per centimeter. So you'll remember a minute ago, I was using electric fields for wave number for megavolts per centimeter. So this basically has units that say how much a spectroscopic transition will shift in wave numbers, which is what IR spectroscopists typically use, for each megavolt per centimeter increment in the field, okay? So the Stark tuning rate is really a measure of how sensitive a vibrational transition is to an electric field. And this is the key to all of our work, is because we need to know how sensitive it is in order to calibrate our experiment. Now, this table, which is a compilation of lots of different things, shows that nitriles, for example, the CN stretch is a strong extinction coefficient, strong at least by IR standards, comes in a very convenient spectroscopic window, especially for working with proteins and nucleic acids, because it's outside of the amide region. And typically nitriles have a Stark tuning rate on the order of 0.4, 0.7 in these funny units. Carbonyl groups, which is what I'll focus on mostly today, have a stronger oscillator strength, so that's great, but they're in this horrible region where it overlaps with everything in the protein backbone, all the amide backbone excitations, and so it makes it necessary to do all kinds of tricks using isotopic labeling, and I'll say more about that later. Stark tuning rate for carbonyl groups is roughly 1. So again, just to make these units clear, if I had a carbonyl group aligned with a 1 megavolt per centimeter electric field and then increased it, let's say, to 2 megavolts per centimeter, it would shift by 1 wave number. It's that simple, okay? The carbon-deuterium stretch is useful in some cases, and if I have time at the end, I'll show you it can be very useful in one particular case. We've done a huge amount of work in this area. Just for the spectroscopist, the change in polarizability for vibrational transitions, unlike electronic transitions, tends to be very small. And really importantly, the direction, since it's a vector quantity, can be obtained by a linear dichroism measurement, and it's typically parallel to the bond axis. So if we know where the CN bond is, even in a very large protein, or where the carbonyl group is, we know where this spectroscopic difference dipole is. Now, nitriles aren't that useful in the sense that they don't really occur, certainly not relevant so much to catalysis, but nitriles do occur on many drugs, and we can introduce nitriles. In fact, Lauren, who's on this call, was part of a group introducing nitriles early on as thiocyanate and also on drugs. And we can also use non-canonical amino acids or modified amino acids. And I won't say very much more about that today. Now, just to make it clear, why are we using vibrations in the first place? Well, electronic transitions are much easier to measure, okay? And they're associated with big molecules, where the vibration is really just associated with 2 atoms. So it's a really small probe, okay? And we're going to be interested in making and breaking bonds, so in catalysis, so it's directly relevant. Our lab and many others have measured Stark effects for chlorophylls, retinals, flavins, and so on and so forth, and they're very, very interesting, but they're not universal. They're very specific. If you're interested in photosynthesis, you study chlorophyll. If you're interested in vision, you study retinal, but you can't put retinal into ketosteroid isomerase and get anything out of it. So we need something that's general, and vibrations are everywhere, basically. I mentioned that the transition moment and delta mu are parallel to the bond axis. That means that if we have an X-ray structure, and we do huge amounts of X-ray crystallography these days, we know what the direction is for delta mu. Of course, IR and Raman spectroscopy are very widely used. There's all kinds of fancy IR time-dependent methods. So this is actually relevant to many areas. There are serious shortcomings. The obvious one is you can't do single molecule experiments yet. It's probably, we'll see. There are some groups trying very hard to improve the sensitivity, but it's usually by coupling to an electronic transition, so it brings back some of these problems. And then there's these background issues that I mentioned that we'll get around. Now, the strategy that we're going to use, I'm going to sort of break this into 2 parts. Focusing on the second part, but let me just mention the first part because this is what we spent a lot of time up until about 5 years ago working on. We were really interested in spectator probes to look at changes in the field. So this is very much analogous to the sorts of things that people look at when they're looking at, let's say, pKa shifts, things of that sort. The first step that I just mentioned is we do a calibration of our probe. And now we're going to make a key assumption, which is that when we put our probe into any environment, its delta mu is an intrinsic property, and that its value is the same for all relevant fields and non-covalent interactions. And I can come back to that if there are questions about that assumption, because it's one that it's probably mostly true. It certainly breaks down in some cases. Once we've measured that, now we can put our probe wherever we want, okay? And then we can make a perturbation, okay? So we're going to change the protein in some way. So we'll call that change the field of the protein. That could be a mute. So I've got a probe over here, and I'm going to look at the effect of a mutation over there, pH change. Let's say if I have a charged amino acid and I want to see how the field is sensed nearby, and maybe compare that with a calculation. Ligand binding, light minus dark, difference spectra, folding. Lots of people are using this now in a variety of different ways, and this is really where we started. Since Marilyn is here, I thought I would just show an example of work. This is work in progress, so I don't really have any results to show you, but just sort of where we're headed with using nitrile. So here's the bacterial reaction center, and here's an example of putting a probe into the reaction center as a spectator. So using the technology of amber suppression for introducing non-canonical amino acids, something we can now do with reasonable simplicity, and even in this 100,000 molecular weight integral membrane protein, we put a nitrile right here, for example. We put it at all these sites that are labeled separately. So this, we take phenylalanines and convert it to, let's say, ortho cyanophenylalanine. We have X-ray structures for all of these, showing that this is well oriented. And then one can imagine doing a visible pump infrared probe experiment in which you actually watch charge as it's moving through here. And so that's a way of actually getting mechanistic information on where the electron is and when, which is still a rather complicated problem in this case, but also on the local dielectric screening inside the reaction center. So this is an example of the use of nitriles as spectators. Now that doesn't really get our problem solved, because our problem is really that we need not differences in the field, but absolute values of the field in order to really understand something about catalysis. And as always, it's much harder to get absolute information than relative information. The way we've approached this, and other groups have as well, is to essentially use another technique for calibrating our probes, but one which we can then evaluate using molecular dynamics simulations. And we do this on a whole range of, using a whole range of methods, and many labs are using different methods, but largely they get very similar results. So what we do is we put our probe, in this case, now we're going to be focusing on carbonyl groups. This is acetophenone, a simple model compound. We put it in solvents of different polarity. I'd prefer not to use the word polarity, but that's a familiar word because polarity doesn't have any units. We'd rather talk about fields. Here it is in hexane, and it's very blue shifted and very narrow. And if we go to more and more polar solvents, this shifts to the red and it gets broader. And this is familiar to everyone from electronic spectroscopy. You take a dye molecule, you put it in hexane, you put it in water, you get the big redshift. When you go to water, there's a whole cottage industry studying that. In fact, that's how solvent polarity scales are calibrated in some cases. There are two things to know. One is that this is easy. This is a trivial experiment. Even the theorists in the audience could do this experiment this afternoon. And the second is, so the shift is big, but also notice this change in line width, which is interesting. And this really reflects the inhomogeneous distribution of fields that the carbonyl group experiences in water. And this is something we can, of course, look at when we do molecular dynamics simulations. Importantly, and this is a note for specialists, we get the same Stark tuning rate doing that vibrational Stark measurement, whether we're in hydrogen bonding or non-hydrogen bonding solvents. And that one of the complications of nitriles is that it's not so simple in the case of nitriles, but carbonyl groups are well behaved. So we're going to interpret this doing molecular dynamics. So we basically take a small molecule like this, put it into a wide range of solvents. Force fields are available for all these solvents. We do either fixed charge molecular dynamics, or mostly now we're doing using polarizable force fields. So using amoeba in one of its generations. And of course, what you get from that is you get a distribution of fields. So just like the carbonyl group in water, you can imagine that the most common situation is that water has there are two hydrogen bonds to the carbonyl group, but there'll be fluctuations with one or the other, or even fluctuations with none, rare fluctuations, or maybe short hydrogen bonds. So you get a distribution in a solvent, something you won't see so much in a protein because of the constraints of structure. But you can take the average value from those simulations, and here then are the fields that come out of such simulations. And as I say, you can do this with a wide range of methods. This is actually quite old work done with Pena Ren, who is one of the developers of the amoeba force field, and Vijay Pandey, but we've updated this extensively. In fact, we have a paper that's just on the Chem Archive right now, if you're interested in sort of extensive testing of force fields in the case of a different, more complicated carbonyl bearing molecule. This particular plot is using a polarizable force field, and so even in hexane, the fields are substantial. I mean, there's electrons and protons in hexane. Remember, I started out talking about 10 megavolts per centimeter. Well, here we are already at 20. By the way, the reason these numbers are negative is because these are all attractive interactions. Okay, and if I have time at the very end, I can come back to that because that's not always the case. And as we go to more and more polar solvents, water being the most polar, these fields get really large. Okay, they get to 70 in these units of megavolts per centimeter. So that's what's plotted on this axis. What's plotted on this axis is just the peak frequencies taken from this simple experiment. Okay, and so I've got the frequency in hexane, the frequency in DMSO, the frequency in water. And what you see when I plot the observed frequency against the simulated field is we get a very nice linear correlation over a very wide range of fields, both for hydrogen bonding and non-hydrogen bonding solvents. And the key here, and I think since I know a lot of people in this audience are doing simulations, the key is that the slope of this line has units of wave numbers per megavolt per centimeter. That's the unit of the Stark tuning rate. We can measure that independently using our vibrational Stark spectroscopy setup, and that gives the same slope. Okay, and so this is actually a pretty nice way to check the validity of the force fields that are used here. Now, once you have this, the concept that we're promoting is that now we take an observed frequency in a defined environment, and from that we can get out the field that's projected onto this probe. Everything here, by the way, is projections, okay, because these are all vector quantities. Okay, so that's really now the key. It's an experiment, it's a calculation. So just as an example, here's a split protein, not of particular interest, but it's a protein that's easy to work with. We have this is ribonuclease S. There we're using acetophenone now as an artificial or non-canonical amino acid. This is made by synthesis on a peptide synthesizer. A small peptide is inserted into ribonuclease that recovers the function of ribonuclease. This is something that was worked out by Fred Richards more than 50 years ago. We've chosen this to be in a very hydrophobic site. And what you see is that when the probe is in water, it behaves as in water. When the probe is on the peptide, it's a little bit shifted. There must be some structure actually here on this peptide, which is itself interesting, though we haven't studied it. And when we put it into this hydrophobic region, it follows your intuition. It becomes very much blue shift. It looks like it's going into hexane or something. It's more like about that on the polarity scale. And also it becomes narrow. Narrowness is important. So at least it agrees with your intuition. So let me just summarize now, putting all these pieces together. We're going to use these field frequency maps, which we get from vibrational Stark spectroscopy and solvatochromism. We combine solvatochromism with simulation, so we get out absolute fields. We get this field to frequency conversion. Then we put our probe wherever is relevant to the problem of interest, measured by IR or Raman, and we can extract out then the electric field sensed by a bond, and then ultimately we want to connect this to biological function. So I hope that sort of lengthy introduction is clear. Let me now talk about a particular catalytic system. This is the first one we worked on, but we've now worked on a lot of others. I just won't have the time to talk about them. So the question, going back to my original picture, is the field from the enzyme actually important? Or is this just, a picture that I can draw, or that Ariel Warshel tells me is important, okay? Ariel has very good intuition, and so, it's worth betting on his ideas. And what we'd expect then is that there would be some connection between the field and catalysis, okay? So we need to actually measure what the field is projected onto a bond involved in catalysis. Now, the enzyme that we're talking about here is a model system. It's not the world's most interesting enzyme, but it's been extensively studied by many careful mechanistic enzymologists, introduced to our lab by Dan Hirschlag, who is a mechanistic enzymologist. And as the name implies, its substrate is a steroid, and it does this reaction. The steroid comes into the active site and engages two very short hydrogen bonds. These are critically important to what follows. These are about 2.5 angstrom hydrogen bonds. And the mechanism of the reaction, which is not in dispute, is a general base plucks a proton off from this position. That produces this dienolate intermediate, which would ordinarily be quite unstable, but the enzyme stabilizes it. And then the general base puts the proton back over here. And so the double bond has shifted from here to here. And so that's what makes it keto for the ketone, steroid for the steroid isomerase. Okay, so the reaction mechanism is not the question here. Transition state lies somewhere here. If you look closely at this picture, you'll see that charge is moving right here at this carbonyl group, okay, as we go to this intermediate through the putative transition state. So this reaction diagram that I started with, which was oversimplified, we can now be a little more precise. The carbonyl group, of course, has some charge separation in the starting material, and we go then to more charge separation as the reaction proceeds. So the difference dipole for the reaction is the transition state minus starting state dipole interacting with the electric field, if it's important. The question then is the field really there, and is it focused at that site? So the good news is that we have a really great inhibitor for this enzyme that's provided to us courtesy of the former governor of the state of California, and that is 19-nortestosterone, which is an anabolic steroid, which is an incredible pain to get into your lab. You have to lock it up, even if you have like 25, you can get it at any gym, you can get large quantities of this, but if you buy it, you have to go through all kinds of crazy things to get 25 milligrams of it. In any case, it goes right into the active site. There are in the literature, we have added many X-ray structures of this bound and mutants and so on and so forth. It engages these same two hydrogen bonds, and you can see the double bond is already there, so there's nothing to do. It's an inhibitor. It's a very good inhibitor. So we look at this carbonyl group. We've made the O18 version of this because, again, we have this background problem. So we have the O18 and the O16 version. We take different spectra to get our IR data, and the IR data is really surprising. Okay, the very first thing you see if you look at the wild type enzyme is that the peak is shifted way far for this carbonyl group to the red. And if you follow what I'm saying, what I mean by red is it's to the red even of where water is. Remember, water had a very big field. That was its average value. It's broad. This thing is way to the red, okay? So that would imply a very large field, okay? And it's also narrow, which is interesting. There's much more to this story. We have a paper which is in progress now that deals with that, but I'm going to focus just on this simplest picture today. This was work done by Stephen Frieden and Sayan Pachi, a graduate student and postdoc. Both now have their own labs. So that's interesting. But of course, it's an enzyme, and we have this long history of other people's work making mutants at the active site. And I'm not going to go into any of these mutants in detail, but just to say that as we make this a worse and worse enzyme, so as KCAT decreases, this peak for the carbonyl group shifts dramatically to the blue. This is not a hard thing to see. These are very large. This is a very large change to the red, and these are large changes to the blue. So there, you just look at this and you can see that there seems to be some connection between the field, if frequency reflects the field, and the catalytic rate, kcat. Now, a legitimate criticism that is often raised, and it is a real problem in mechanistic enzymology, is that when you make site-directed mutants, which are easy to do, you're really changing the amino acids, and it's hard to change amino acids without changing things. And we're very sensitive here to local structure. So we want to make changes that are as modest as possible. And for that, but of course, if they're modest, then the effects are going to be smaller, but nonetheless, we can measure them. It turns out that this, I focused on this tyrosine and aspartic acid, both making these short hydrogen bonds to the relevant carbonyl groups. It turns out there's actually 3 tyrosines, and there's a whole story that's associated with these 3 tyrosines and the pKa shifts, very anomalous pKa shifts of this tyrosine cluster, which is an interesting story in itself. What we do is we make at each site independently, we put in chlorotyrosine, monochlorotyrosine. So if we put it at this most remote tyrosine, you see that the peak shifts a little bit to the blue relative to wild type. And in fact, we measure the rates. The rate gets a little bit slower. We put it there, there's a bigger perturbation. We put it at this site, there's a bigger perturbation still. Here's a double mutation combining position 57 with this single site mutant that I had on my last slide. See, there's a systematic progression to the blue associated with a decrease in kcat. And the structures here, at least within the resolution of, these are reasonably high resolution structures, are unperturbed. So that's important. So now we put all this together, and what we have is from our, if we go back to our picture here, we have, frequencies. We have our frequency to field calibration that I described before from solvatochromism and the vibrational Stark effect. And so that gives us the sort of this axis, which is sort of a combination of experiment and these simulations. And then on this axis, what I'm plotting is the activation free energy. And the activation free energy is coming, in this case, from transition state theory. So I'm doing what mechanistic enzymologists typically do. And in fact, this was the range I was talking about before. Wild type is about 11.5 kilocalories per mole, and we make it worse and worse and worse. So this is the fastest enzyme, this is the slowest. And if we now plot so these are independent measurements done by many other labs, and we've repeated them all, and they were carefully done. This is a measurement of rate. This is a measurement of field through these IR frequencies. What we see is when we plot these, we get a very nice linear correlation. So this is what physical organic chemists would call a linear free energy relationship, except that unlike any linear free energy relationship in the literature, here this axis means something in an absolute way. These are absolute numbers. These are electric fields. You can calculate these things from first principles. Now, what's interesting to us is not so much the slope here, which actually gives the delta mu of reaction. I don't want to focus on that. What I want to imagine is something we can't do experimentally, but we can do with this data, which is we can imagine shutting off the field, that is extrapolating to what the activation free energy would be in the absence of electrostatics. That's the y-intercept, in this case 18.4 kilocalories per mole. And that then brings us back full circle to where we started, because now we have an actual quantitative measurement of what the contribution of electrostatics is, or electric fields, to the catalytic proficiency of this enzyme. So you remember I started talking about 22 is the uncatalyzed reaction in solution, and it's about 11.5 in the enzyme. And what we're finding is about 18.4 of that, or 18.56, something of that sort, is due entirely to electrostatics. And so in this case, that would be 70%, okay? So we have a quantitative picture now. So it's not 100% like Aria wanted, but it's a substantial contribution. And as I said at the beginning, what you find in most biochemistry textbooks is the rest of this stuff, but at least for this enzyme, that's actually not the major contribution. It's not no contribution, it's just not the major contribution. Now, I think, you know, I went into this unprejudiced. I just wanted to know, you know, can we do this? As I said at the beginning, there are very strong feelings about, you know, what the relative roles of these things are. And there's nothing magical about 70%. This is what it is for this enzyme. We've looked at it turns out there's lots of data in the literature, mostly Raman data, beautiful data where people have made mutants and they've looked at rates, and they've even noticed that there's some connection with what they thought was the polarity of the changes. And we've taken all that and reinterpreted it now, having calibrated the probes. And in fact, the numbers, you know, for the contribution of electrostatics vary all over the place, not too surprisingly. It depends on the system, but the same concept or approach can be used. So this is, I think, a fairly general approach. There's another side to the whole story of electrostatic catalysis, which I haven't emphasized. I'm only going to talk about it very briefly. This was published recently, and it has to do with electrostatic pre-organization. It's again a concept very much promoted by Ariel Warshall. And just to make a long story short, I've been playing a little fast and loose with my picture here because I've essentially assumed that these vectors that everything was parallel here and that one field applied. But of course, there could be a different field associated with the transition state as opposed to the starting material. Now, we can, of course, take that into account. These are all just vector changes, but now we have a problem. How are we going to get this information? And as you can imagine, because of rehybridization at that carbon, this is going to, one might expect a fairly large geometry change. So just to sort of give this sort of one slide quick view, we used a substrate-like analog, which has an sp3 carbon here, and then 19-R-testosterone, which is more like a transition state analog in the spirit that I introduced it. And then looked at X-ray structures, reasonably high resolution X-ray structures. The overall structures are identical, but if you look at the that carbonyl group, the enzyme actually is pushing that carbonyl group both here and here towards a transition state like geometry. And so in fact, we were getting away with murder here basically because it turns out the field in this case is pretty much the same, roughly, in the starting material and the transition state. So there only are very small geometry changes due to this distortion. So just summarizing then all this story about this particular enzyme, just to sort of focus on it, and then I'll just talk very briefly about some other things. These vibrational Stark effects are a quantitative and directional probe for electric fields in proteins. And we require solvatochromism and simulations to get absolute fields. The simulation part of this is a really important aspect because it turns out there are many different ways of simulating these fields. Most of them give very similar results. And we're doing, right now, working with Tom Markland's group, we're doing some really elaborate, fully quantum mechanical simulations of everything. So this is a quite complicated story, but we're getting actually, even there, very similar results to these classical molecular dynamics, or at least when using polarizable force fields. In the case of KSI, we see a direct connection between electric fields and the activation free energy, and from that we can extrapolate what the electrostatic contribution is to catalysis for that enzyme, and we've done this with others. This concept of pre-organization is important, and again, this will be very familiar to someone like Marilyn or anyone who comes from the electron transfer field, because it's fundamentally different from the reaction field, okay, which is really optimized for the ground state configuration or ground state charge distribution, and has to reorient to accommodate the transition state as a reaction proceeds. You may have noticed the fields in water are very large, and yet the rates in water are often very, very slow. And that's really what's limiting the catalytic capacity of water, despite the significant magnitude of its electric field. And this is, again, a concept that Arya has described in many papers, but has not been, I think, fully appreciated. Now, once these results were published, and we've updated them on several occasions, we and other groups did calculations. What are the fields? And in fact, they get results. Teresa Head Gordon's group, Tom Markling's group here at Stanford, and several others, including ourselves, get results which are actually in pretty good agreement. And the nice thing, of course, about simulations is now we can look at the contributions to the field, and it really is those two short hydrogen bonds that are the dominant contribution to the field. And I should add that, you know, classical force fields don't like those short hydrogen bonds, and so you have to go to pretty sophisticated methods to get around that. And Teresa Head Gordon in particular, I think, has really led the way on that. I should note, because I'm often talking not to a biological audience, and of course, I think these concepts also apply to non-biological catalysts, and that's, you know, again, there are these vague references to electrostatics, but I think now this is going to, you know, now many labs are starting to think about this. You know, enzymes aren't magical. Now, I think, Walter, I'm running long here. Can I just maybe say just a few quick words about these topics? But this is going to be so quick that it'll be embarrassing. I think I just give you a flavor, so the next time we meet, you'll know what I'll be talking about. Okay.
We have time. We have time.
Okay, and you can hold me. You can hold me to it. Okay. So I'll talk briefly about the evolution of fields. Can we get more information than just one projection? And then I'll only very briefly mention this excited state catalysis thing. You can read about it. So I showed you this picture of ketosteroid isomerase, and an obvious question is, well, you know, wild type is wild type, but if I made a better ketosteroid isomerase, a better catalytic, would the field be larger still? We thought a lot about this and how in the world you would select for a better ketosteroid isomerase, and others are now working on this. Hopefully they'll come up with something and we'll be able to do the measurement, but it's kind of a clumsy reaction, actually. But of course, there are lots and lots of examples of enzymes where we have information on their evolutionary history, or by directed evolution, we can look at their evolution in the lab and ask about how they've evolved. So the system we've chosen to work on, I'm not going to tell you anything more than just sort of give you just one quick slide on results, are the beta-lactamases. And the beta-lactamases, of course, are the enzymes responsible for dealing with antibiotics, and the history since the introduction of penicillin of the evolution of antibiotic resistance, of course, is the history of the evolution of these enzymes, starting with what's called TEM1, which was back sort of the first sequenced example, up to, let's say, TEM52. There are hundreds of these. This is one whose pathway evolution, now evolution driven by in the clinic, have been characterized in detail. And so we go from an enzyme that, you know, is great at dealing with penicillin to one which is terrible at dealing with penicillin. That's what antibiotic resistance is. And now we have sort of advanced second and third generation antibiotics like the cephalosporins. Here's CTX, which is just one of the hundreds and hundreds of examples. So this goes from being a sort of a narrow spectrum beta-lactamase to a broad spectrum beta-lactamase. But here we have an example where KCAT for one substrate is going down, KCAT for the other is going up. And if our hypothesis is correct, we might expect the field to evolve in parallel with this. This is sort of evolutionary time. The story turns out to be much, much more complicated, and that's why would take me an entire hour to talk about. But just because I think this is so cool that we can actually, when I say we, graduate student Sam Schneider, postdoc Jacek Kosic, what we can do is we can actually look at these enzymes. We have to now make C13 labeled, since we need labels on this beta-lactam. That's of course the key carbonyl group that's involved in the hydrolysis of penicillin by beta-lactamases. We can make a C13 version. We put a huge, reason I'm showing you this is this was a year of work, okay, a very hard work of growing mold on purpose in the laboratory. We grow mold all the time by accident, but this was a case where we had to grow mold on purpose. You need special rice, you need special sauce, all kinds of magical things. It's an incredible pain in the neck to work on. This heroic effort from these two guys was published last year. We actually learned something about the penicillin biosynthetic pathway in 2020, which is kind of amazing. Now, the solvatochromic calibration of penicillin turns out to be a huge story in itself, much more complicated than acetophenone, because there's this side chain here, and that has multiple conformations, and there's a charge group here, and the two of those things conspire to differently organize the solvent around this carbonyl group, and that has rather profound effects. If you're interested in that, that's just been uploaded a few days ago to the Chem Archive. And we'll see how reviewers deal with it. And then we can put this into a whole series of evolved enzymes. And what you're seeing here these are different spectra. So we have C13 minus or C12 minus C13 different spectra. And what we can see is that actually in some cases the field doesn't evolve, and in other cases it does. And that's a very complicated story, which I'll leave for the next meeting. But we have a huge body of data, now a huge body of simulation, and we're in the process of, we'll see, this is being submitted, and we'll see whether reviewers find it interesting or not. It is actually a very interesting, we know much more about the kinetics of these enzymes now than was known before. This is a field where there's an enormous amount of work, but very little of it is very physical or mechanistic. It's pretty frightening. Okay, that's one quick story. The second quick story is electric fields in 2 dimensions. So this is also some work that's in progress, and this is really interesting, and it's the work of a graduate student in the lab, Chu Zhang, who chose this because this is an enzyme that has zinc and its active site. He was specifically interested in the electrostatic contribution of this metal, and I don't have anything to say about that today. It, of course, is involved in the conversion of alcohols to aldehydes. It's reversible, and that's what attracted us to it. Because of reversibility, that delta mu of reaction in the forward and reverse direction should be different. The transition state presumably is the same, but the dipoles and the reactants and going forward and in reverse are different. And so that's what one of the other things that attracted us to this. Now, if you look at the substrate here, in the case of this is an inhibitor, which is an aldehyde. There's, of course, the carbonyl group that we're all familiar with and we've now seen many examples of, but there's also a carbon hydrogen bond, and we can replace that with deuterium. And now we can look at the CD stretch and the CO stretch simultaneously so these are roughly at 120 degrees at the active site of this enzyme. And there's right away a real surprise here, and that is, and again, this experiment could have been done many years ago. If you look at the carbonyl group on aldehydes, they behave just like what I've described for acetophenone or that Schwarzenegger inhibitor. But the aldehyde CD stretch goes in the opposite direction. It moves actually to the blue as we go to more and more polar solvents. So this is a very surprising result. And our intuition on this is that the solvent is organizing predominantly around the dipolar carbonyl group. And because of that, it's creating a destabilizing field at the carbon deuterium bond. Okay, that's a sort of a very qualitative picture. It's borne out by simulations, and we're doing this at every conceivable level. This is work very much in progress. What really motivates this is kind of a stunning result. If we put this inhibitor into the wild type enzyme, and of course we have many mutants and metal variations and so on and so forth. This is just wild type. The carbonyl group just shift very far to the red, not as far as in so the fields aren't as large as they are in ketosteroideoisomerase, but the CD stretch shifts way to the blue. It's just completely in opposite direction. So this really now will be a challenge for theorists to compute these fields, because now you have, it's not just one projection, which you can do all kinds of things with, but when you have 2 projections, you're much more constrained. So we're very excited about this. And the last thing, which is I'm just not going to talk about, okay, because it's been published. It was published last year in a paper in Science, and there's much more. We're just submitting more papers on this now. Has to do with something which you might find to be a completely unrelated problem, but it turns out that in the excited state, many molecules which undergo isomerization reactions, that isomerization reaction also moves charge, okay? And that can be shown in simulations and has been shown for many years. And in the same way that I've been talking about here, the differential effects of the protein environment on which bond rotates, let's say in retinal, or in the case that we looked at in GFP, where there are two possibilities, the relative activation energies for bond rotation actually can be shown to depend on electrostatics. Again, this was done in a rather qualitative way using amber suppression to modify the chromophore and now the environment. But that's a long story, and if you're interested, the first paper on this was published last year. But you'll see that the language is exactly the same as the catalysis language. So we think this is really a broad way to think. Okay, I've come to the end. Let me just quickly wind up. A lot of these people are no longer here. In fact, of all the people on this slide, the only person who's currently in the lab is here. This is work done by Nick Levinson, was one of the first people who started the vibrational sabatochromism work, a person that Lauren knows very well. I think she may have had something to do with attracting him to our lab. He's now got his own lab at the University of Minnesota and is doing great. He was working with Stephen Fried, who was a gifted graduate student, and a postdoc, Sayan Bachi, who came from Robin Hostrasser's lab. Both of them now have their own independent lab, Sayan in Pune in India, and Stephen at Johns Hopkins. They were joined then by Yufan Wu, who's now a postdoc at Harvard Medical School, working the experiments I showed on chlorotyrosine and electrostatic reorganization, which I mentioned only briefly, was really Yufan's work. These other enzymes that I very briefly touched on at the end, you've already seen these two pictures of Sam and Yatsik, and here's a picture of Chu, who's doing really interesting work and has now been joined by a whole bunch of other people. The GFP work I didn't mention were done by these folks. And this work has been supported pretty much for a long time by the NSF and NIH. I'm very much indebted to them for that support. So thank you, Walter, for letting me be a part of this community. I hope this was relevant to some of you.
Thank you, Stephen. That was a very interesting and deep presentation. And I'm eager to have you here when the situation will become this possible. So we can start with questions, of course. And before, okay, I already have one question from Christine Paul. No, I don't know. Okay, that was close. Let me ask something first. Do you think there is some computational advancement or tool or new way of helping the experiments you are doing in order to interpret them or to provide some help?
I think the answer is yes. I mentioned that the classical, just for sabatochromism, classical molecular dynamics works quite well. In fact, the Onsager formulation of this problem works quite well. That goes back only 70 or 80 years. The problem with the Onsager formulation, for people who are familiar with it, is it involves a cavity whose radius is a variable parameter. But if you make reasonable guesses, you get results that are quite similar. So the fixed charge molecular dynamics and polarizable force field using amoeba give similar results. Where things get really nasty, and that's why I wanted to put this back, and I would ask you maybe to go through this paper, because that will maybe help to answer your question, Walter, because the answer is not simple, is that it's really easy to do these calibrations on simple molecules like acetophenone or acetone, or even these aldehydes are relatively simple. But as soon as you start getting a little bit more complexity in the molecule you're interested in, and penicillin is a perfect example of that, where you've got that side chain and you've got the charged carboxyl group right very, very close, then you get very weird results in solvatochromosm, and the simulations get very strange and complicated. And I think if you look at this paper, we've gone through essentially everything that's available, including 8 different water models, for example, and we can make improvements, but we don't really have we can't just say this is our go-to method. Let's just do this, and we'll get the answer. And so if you guys could come up with that would help. The harder problem, I think that's a hard problem. But the harder problem is really doing this inside the protein. That is, what level of simulation do you need? So for example, when we started doing simulations, we're not primarily a simulation group, obviously, on KSI, what we found was that, not too surprisingly, the force field didn't like those short hydrogen bonds, which are seen in the X-ray structure. And so it just pushed the steroid out, and the fields dropped, okay? And so you really have to hold it in place. So you need a little more information than you'd like in an ab initio method. I would urge you to look at papers from, Teresa Head Gordon has published now several papers on basically motivated by this, and now generalizing it and testing it, actually. She's done some, a little bit of experimental work. But I think that's where the real challenge is. But I can't tell you what the magic solution is. Todd Martinez, my neighbor, would say, well, just do 10,000 atom full quantum mechanics on the entire thing.
Couple cluster.
Yes. And we have 500 GPUs, and let's just do it. And we've done that, by the way, and on some other problems we've worked with Todd, and I don't think Todd is here, so I can say, what you get is you get a number at the end, but you can't really tell what the hell happened. The nice thing about these force fields is that you can these classical pictures, you can go in and look at things, and you can sort of, but it does bias what you do, because you sort of know what you're looking for. So I'm not answering your question.
Okay. Yes. Okay.
Maybe I am answering it.
We have a question from Marilyn.
Yeah. Hi.
All right. Well, I always have a million questions, but 2 simple ones. Why? It would seem to me that the hydrogen bond strength would really be important, and you said that the shift isn't dependent on hydrogen bonding. And the other thing is, when you look at the fields in molecular dynamics, you show like nice little dots. What's the standard? How much are they fluctuating?
Yeah. Well, first of all, what you said first is not correct. The hydrogen bonds definitely affect the fields. What they don't affect is the Stark tuning rate, delta mu. And so, and we can show that by a number of methods. So when we first presented this work, we were severely challenged by let's say the descendants of the Jenks perspective, who said to us, well, if you have a red shift going from, let's say, hexane to water, these people come from sort of an organic chemistry mindset. They draw resonance structures, and they would say, well, something goes to the red, it's more like a single bond, and so you're stabilizing something that's more like a single bond. And the word that's used by some in the mechanistic enzymology literature is bond polarization. Well, first of all, you can calculate what the effects of bond, how big those would be, and it's really small. But we did an experiment, because if the bond is becoming more like a single bond, then you would expect the anharmonicity of the bond to change a lot. So we got together with Marty Zanni, who does 2D-IR. 2D-IR is actually a very useful way to measure essentially overtone absorption. So the lowest vibrational level to the first, but also the first to the second. And that's, of course, how we measure potential energy surfaces. is by measuring overtone absorption and harmonicity. And we did that, in, I think, hexane, DMSO in water, where there's a big spectral shift, and the anharmonicity was unchanged. So the bond doesn't change, okay? At least within the accuracy of that measurement. And that, but it was a very carefully done measurement. So we can also measure the vibrational stark effect in a mixed solvent where we have both a hydrogen bond and a non-hydrogen bond population. And now in an external field, the vibrational stark effect is the same. So there seems to be no effect. This is something we've worried about a lot. It's important. Now, I've actually forgotten what your second question was.
What's the fluctuation in the field?
Yeah, well, that's a huge question, usually interesting. So in the case of simple solvents, we actually have information on that because the origin of the line widths is largely inhomogeneous. So in water, you have that very broad line, and hexane relatively narrow. But we know from measurement of vibrational lifetimes, we know what the intrinsic line widths are, and they're narrower than that. So there's inhomogeneous broadening, and it's much greater in water than in hexane. And of course, in our simulations, we get that as well as the average position. And if we plot, same way I plotted the frequency versus the field, if we now plot the line width, or a measure of the second moment versus the field, we also get a nice linear correlation. Not quite as nice as what I showed, but a pretty good one. We haven't pursued that very much, but it's a really interesting observation for people interested in solvent-solute interactions. What's much more interesting to me is why these bands are so narrow in the protein. And if you step back and think about it, there'd be nothing to talk about if the bands weren't narrow. It'd be impossible to measure. If it was as broad as it is in water, even our best difference methods would be very hard to measure. The question is why, let's say in wild type in KSI, is it so narrow? So there are sort of two limiting views of that. And this is, again, maybe Walter, your question. One would be that the active site is associated with pre-organization is relatively rigid. That is, the fluctuations are damped somehow. The other is exactly the opposite of that, which is the fluctuations are so fast that what you have is effectively motional narrowing. Okay, so those who come from a magnetic resonance background will be familiar with the concept of motional narrowing. That's something that also applies in IR spectroscopy, but of course the time scale has to be really fast. And this is a perspective put forward by Lu Wang, who was a postdoc in Tom Markland's lab, and when we worked on some of these simulations. And Lu is now at Rutgers. And what we need is, this is a case where doing a dynamical measurement in KSI would be good. We tried, but the background, unfortunately, for 2D IR was just too large. Now maybe that will get better at some point, but it's a really interesting question, Marilyn. You should appreciate there's a huge literature of IR and Raman spectroscopy in proteins, and you might ask, well, why can you see anything? It's because the bands are narrow, almost always. And the question of why is a really interesting question.
There is a question on the chat from James Meyer. Is there a fundamental difference between describing this as a vector electric field rather than the sum of electrostatic energies? If the delta mu is the same, are these equivalent?
I'm sorry, you're going to have to say that again.
I can say it.
Okay.
Steve, this was a fabulous talk. I think that your dichotomy between the Jenks picture and the Warshall picture is very interesting and stimulating, but the field that you have comes from electrostatic positioned partial charges and things like that. And could you just sum up all the energies and not deal with a field? Is that conceptually the same, in which case the Jenks, the enzyme has positioned the charges, or is it conceptually necessary to have the vector field and not just sum the energetics of the individual electrostatic interactions as a delta G at the transition state?
Yeah, that's a great and very nasty question. No, you're absolutely right.
It's meant to be friendly.
No, I understand. No, because of course that's exactly that hybrid view, let's put it that way, is I think the right way to look at it. What I set it up as a polarized view because the people in the field have set it up that way, okay? And I think that what you're saying is really the right way, because it is precisely the organization of, using Arya's words, I mean, the organization of the dipolar, polar, and polarizable groups. That organization is what Jenks was talking about, that gives rise to the field. But if you really want to understand at the end of the day what's responsible for lowering the activation energy, you have to have some metric of what happened, okay? And I think the field is the metric, or at least the field is one metric, or a metric for that part of the catalytic proficiency, which is due to electrostatics. Let's put it that way. So that's I think the spirit. But I think we don't want to just have this come down to semantics. I think it's a little bit more subtle than that.
Okay, thank you.
It's a very good question.
Then we have a question from Daniel Konstantinowski. When he says, you mentioned that water doesn't organize to promote the transition state in the bulk. Have you ever seen a case where semi-structured water at or near at an active site strongly participates in the transition state promoting electric field? And can you imagine enzyme harnessing the electrostatics of structural waters to stabilize the transition state?
Absolutely. And in fact, this is an example. It's right here. It's just that you can't see it. I mean, maybe I can actually blow it up. I don't know, is that working if I blow it up?
Yes.
Yeah. So in fact, it's critical to the function of the sort of second step. I mean, there's an acylation step and then a deacylation. I didn't go through the mechanism here at all for the beta lactamases. So here's the initial acylation. By the way, this is involving this serine. The strong hydrogen bonds here, we think, are due in this case to amide backbone NH, much more tricky to modify than was the case in KSI. But the second step is this water molecule, which is seen by X-ray crystallography, is activated by this neighboring glutamic acid. And that is responsible, in fact, for that. It's the water molecule that attacks the acyl enzyme intermediate to produce the hydrolyzed product. And in fact, in these experiments, we're forced to modify that glutamic acid in order to actually see something, because otherwise we're looking at a substrate here, not an inhibitor. So the answer is absolutely yes. And by the way, there are many examples where there's a water at the active site of an enzyme that's seen and often seen in X-ray structures and is critical to function. I think, does that answer the question? I think it does.
Okay, then I have a question from Thomas Simonson. Thomas, do you want to make yourself?
I can try to make it myself if my microphone is working okay.
Yeah, I can hear you.
Okay, good. Hi, Steve. It was absolutely wonderful talk. Very, very impressive. My real question is kind of, multiple and kind of complicated. And what I would really like to discuss was these ideas of pre-organization you showed so beautifully. And just go a little bit more into detail. I think that'd probably be hard to do in this particular framework.
Yes.
So I've tried to boil it down to just a very, very simple point to kind of test my understanding of some of the things you showed. I believe what I understand is you're probing the field on particularly these two active site tyrosines, which are just adjacent to the hydrogen bonding tyrosine. And you mutate them with this non-canonical amino acid, and you measure fields. And so, is it correct that you're measuring the field on these two particular carbonyls, and you're getting not exactly the same, but similar result for the field at these two active site positions? Is that correct?
Yeah, it is more complicated than that. So first of all, we're not measuring the field of those two amino acids, that aspartic acid and tyrosine. We're measuring the field created by those two as they interact with that carbonyl group. And what we have is we have a carbonyl group in the, in one representation, which is sort of reactant-like, and the other one is transition state-like. And this, by the way, is discussed because this was published last year in a paper in JACS, and it's complicated, very, very complicated. And basically what happens is that the enzyme is distorting both the ground, the starting material structure towards the transition state geometry, and the transition. And the interesting and surprising thing is when we did a high resolution structure of 19NT bound at the active site, it's also distorted, and again towards what we would calculate to be the transition state geometry. And therefore, we would imagine, we obviously can't see transition states, we would imagine that the fields would be roughly the same then for both. And that was assumed in the way I did the initial analysis, right? I just factored out the field, and I took the field times the difference dipole, but actually the field could change. And in many enzymes, I imagine it does change. That's a really challenging problem. Then you need to do two experiments, or at least two experiments. So I would oh, I can see you now. Hi. I would send you to that paper. It was published, I don't know, mid-late last year in JACS. And I think it goes through the details, because there are many calculations in there. There are multiple structures, and there's gas phase. I mean, it's complicated.
I'm going to go back and read your paper carefully. Maybe just as a final comment.
Yeah.
I think the 70% number might even be a bit low. I would have guessed an even bigger number for electrostatics, but that was a wonderful talk, and I really liked it. Thanks very much.
When you say you think it's low, I mean, it is.
Well, it's not low, it's high. Of course, you're right, it's very high. It's just that I would probably have guessed even higher.
For specifically for KSI.
No, just as a general idea.
Yeah, well, we published a paper basically where we didn't have to do anything. We just took other people's data. We had to recalibrate because they weren't thinking about it in the way that we're thinking about it. And we looked at some chlorodehyalogenase, and I think there the contribution was about 55%. And I don't think there's nothing universal about this. I mean, there are other effects that are at work that are not primarily electrostatic. And Arya wants it to be 100%. He was very disappointed. So I think he likes that at least it's not zero.
Thank you.
Okay, I have a question from Eleanor Stewart-Jones. She's saying, when you are just looking at this solvatochromism of a molecule, to my understanding of your talk, you look at how the vibration of a specific bond changes in a range of solvents. But don't proteins have to be kept in a buffer? Does the presence of the buffer complicate how you interpret the electric field felt by your probe when that probe is in a protein? Please let me know if I misunderstood anything. Thank you for the talk.
Yeah, no, I mean, presumably when these, take ketosteroid isomerase, that's a really good example. That binding pocket is really, really deep inside the protein. And in fact, when the substrate or inhibitor binds, water is entirely excluded from the active site, water and the buffer. So presumably the buffer is affecting something global. We haven't looked systematically at whether, for a probe buried deeply inside a protein, whether buffer conditions actually matter. At least we haven't looked at it intentionally. We probably have looked at it without thinking about it. I don't think I would be very surprised if there were an effect, at least over the reasonable range where the protein, pH range where the protein is stable. There's a really interesting story. I know probably Marilyn has heard this story concerning that the enzyme, when you don't have a substrate there, and that tyrosine at the active site. This is work that was initially work we were doing with Dan Hirschlag, and it's, you know, since a lot of people are interested in pKa shifts, it's a great example of where things can really go wrong. You know, there's this oxyanion hole, and you ask, well, what is titrating at the active site? And, you know, what we did was we put probes all around the active site. These probes were thiocyanate. So you make well, there's no cysteines. You take the cysteines out in a protein, put thiocyanate in, typically in the place, let's say, of a methionine. You can pretty much predict exactly what the orientation will be. And we have structures for all of these. And then you use the nitrile as a probe for, let's say, doing a titration at the active site. Now, this is without a substrate present, okay? So, you know, there was known to be something that titrates in the active site, titrates around 6.3. And so we said, oh, it's, you know, in the aspartic acid or something like that. But, you know, it turned out that none of our nitriles, they were all shifting the wrong way in response to the pH change. So we happen, for reasons, again, Marilyn will know why, we happen to have C13 labeled tyrosine in the lab with a C13 right at the hydroxyl group. And we put that in to there are 4 tyrosines in KSI. And one of them is on the surface and behaves normally. And remarkably, that cluster of tyrosines has a hugely shifted pKa, and it's really an example of, you know, something that isn't in a simple, you know, acid-base kind of consideration, because what you have is essentially a proton that's delocalized in this cluster of tyrosines. It's, again, Marilyn will be familiar with it. It's a little bit like the redox potential shift of a special pair in the reaction center compared with a monomer. When you delocalize, you get a shift. It's not something that's in conventional pKa calculations. And there's a lengthy paper on this written in collaboration with Tom Markland's lab, because among other things, there's an anomalously large equilibrium isotope effect on that pKa. And so we were really puzzled by this, and working with Tom and his postdoc Lu Wang, we were able to, I think, model that reasonably. That's all for the apo protein. Once the substrate's there, the water is gone. I don't think the buffer matters. Going back to the original question.
Okay, I have a question from Jim Warwicker. With regard to directed evolution for enzyme activity, is it fair to say Kn equal shape, easy, and Kcat much more complicated?
That's a general question. That's not something I've yes, I think that's fair to say. I guess I mean, we don't know experimentally. That is, we haven't yet looked at a library of proteins evolved by directed evolution. It would be really interesting to do. In principle, that could be done with the beta lactamases, because there's a 5,000 member library that was made by Rama Ranganathan of beta lactamase mutants, but we haven't looked at it. So I really can't answer the question experimentally. And I would prefer to do an experiment before and know what the answer is before answering.
Okay. Then I think that Thomas Renger wants to ask a question.
Hi, Thomas. Hi.
Hi. Thank you very much for this great talk. I want to come back to the broadening of the vibrational transition that you mentioned, that in the protein they are so narrow, and you had 2 explanations. One is motional narrowing, and the other is that the protein somehow reduces the fluctuations at the active site. So my question is, could you check the temperature dependence of this broadening? Because it should go in the opposite direction, the 2 explanations, or?
No, that's a great suggestion. It's a hard, it would be a hard experiment to do. But you know, you might know, I mean, there's nothing unique about KSI in this regard. This must, I mean, and in fact, Raman would be a better way to do this. I mean, haven't people done experiments like that? I mean, do you know? I mean, I turn the question around.
No, I haven't followed this vibrational spectroscopy. I'm working on the electronic spectroscopy only.
Yeah. Yeah, but you know, but I mean, there are thousands, literally thousands of. I mean, there would be no Raman literature and IR literature on proteins if the bands were all as broad as they are in water. And you know, what I presented were just two limiting models, I would say. And I don't know. We really don't know what the answer is. There are. You're right. The temperature dependence would be one way. Temperature is, as everybody appreciates, a really complicated variable because lots of things change when you change the temperature. And I'm not sure you'd be able to specifically target that. But there are these nice, you know, vibrational coherence measurements which provide direct information on this. And I'm hoping that, you know, somebody will do that. There's been, I mean, since I've been talking about this over the last 2 or 3 years, there are people who have sort of grabbed on it. There's some theoretical papers, some work from Arizona State just came out on sort of, I would say, speculating on why this might be, but it would be nice to see some experiments. I'll tell you, Thomas, I find temperature to be a really complicated parameter, but I understand why you're suggesting it. It's a good suggestion.
Is it in principle possible to measure it, or are there some experimental difficulties?
No, I mean these measurements. I mean the experimental difficulty of these measurements really is that your carbonyl group is in the midst of this huge background absorption from the amides or H2O or D2O, and we do everything we possibly can to minimize the background. But for example, those spectra I showed just briefly, and I'll talk about next time maybe. These were painful experiments to do because you can see the difference. By the way, you'll notice that, and you can see it right here. It's a perfect example. If you look at the difference spectrum in water, there it is. You can see that the positive and negative are almost canceling each other because both of them are broad. Here it is in the protein. Boom, boom, completely separated. Okay. That's a perfect example of what we're talking about. We only on the bottom of each one of these plots are showing the actual difference spectrum. Boom, boom. The isotope shift is really the giveaway. If we didn't have this isotope shift, there would be this enormous background that you would have a very hard time dealing with. That background itself will be affected by temperature because it involves water, it involves all of it. So that's the kind of thing that would make this a very.
I see the difficulty now. Yeah.
Make it a very painful, make it a painful experiment.
Yeah.
I'm trying to imagine trying to persuade somebody in my lab to do this. I think they would rebel.
Thank you very much for this very nice talk. Yeah.
Thank you.
Wei Yang has a question.
Yeah, just follow up what Tom asked regarding the 70% contribution from electrostatic effect. I think that's really touched to a topic in theory people have been talking about for a long time. It's called free energy decomposition, right? Free energy in theory cannot be decomposable, and so decomposition depends on the pathway. And so I think the way in history people have been justifying the way, if I have one way to decompose, right, I need to define the state. I need to give some meaning about the state, right? My question is about actually this your zero field state, right? Because you do extrapolation and you have this zero field state. So can you tell us your insight about this assumed zero field state, right? Why it is a good way for free energy decomposition?
Sorry, I don't understand what you mean by zero field state. I mean,
right. I mean, in your case, you have extrapolation, right? You have a mutant, and then you intersect 18.
Oh, I see, the zero electricity. Okay, in the end, I thought you meant zero field in the solvatic chromism because, of course, there what you extrapolate to is the gas phase frequency, and in fact, that's what you extrapolate to. And the gas phase frequency has been measured for things like acetophenone, or it turns out even for penicillin. We were amazed to discover recently. But the extrapolation that's an completely made up extrapolation. That is, we extrapolate to something we could never measure. And you could argue, and some people have, that extrapolation is just that. It's it's an extrapolation, and so it may be that there's actually some funny business happening, and that's wrong. But that's what we did. Okay, we simply took that line and ran it to its y-intercept, and the y-intercept is what the activation free energy is in the imagined absence of electrostatics. And there's no decomposition there. I mean, the decomposition, I mean, you could say that these mutants are in effect doing a decomposition. I mean, we're looking at the effects of each one of these mutations. But I think the really best way to look at it is, let's say in Teresa Head-Gordon's work or in Tom Markland's work, look at the computational decomposition of contributions to the electrostatics, which is something we really, I mean, we can't do. I mean, we've motivated them to do that by the experiment, but we can't do it as well as they can. I feel the same way, by the way, about even the solvatochromism. I mean, you know, the beauty of simulations is, I don't explain to this community since many people here do simulation, is you can look at snapshots of the simulation and see what's going on. Whereas in the experiment, we just get the average, okay? And that's just the way it is.
Yeah, right. Thank you. Indeed, actually, there's still a decomposition because there is a assumption in this extrapolation,
Yes.
right? You assume actually your protein structure still hold, right, without electric field.
Absolutely, absolutely. Right. We're just extrapolating. Right. By the way, we're not extrapolating very far because some of those mutants are pretty bad. But you know, there is again this. I think this is a very legitimate criticism, something that Dan Hirschlag often raises as a criticism, but we're all stuck with it. And site-directed mutagenesis is a pretty, like a sledgehammer, you know? If you change a tyrosine to a phenylalanine, you could imagine a water molecule, somebody was asking about water molecules sneaking in. We have structures, so we don't think that's happening in this case. But that's why amber suppression is nice, because it allows you to do more like the physical organic chemists would do, you know, make a more gentle change. Of course, you make a more gentle change, you're looking at smaller changes, smaller effects. And then people will say, well, the effects are too small to take seriously. So it's, we're stuck. That's just the reality of molecules.
Thank you. It's a really stimulating talk.
Okay, well, I'm glad.
There is a question from Chad Isaac Drexler.
Mm-hmm.
Hi, hi, Steve. Nice talk. I am actually curious. So I had written this question in the chat before you just took that last one, where you mentioned that when you extrapolate in the standard Onsager experiment back to what should be the gas phase frequency for acetophenone and all these different solvents, right?
Yeah.
So you put it in all the solvents, you do an MD calculation then to get the field and create your calibration curve. Does that y-intercept you get actually match the value for the gas phase?
It does, but only if you do the amoeba simulation. So if I go back to that slide, actually, this is one of the nice things about having the slide view right there. So this is a pretty, this paper, which is a couple of years ago now, discusses exactly your question in great detail. Okay, because we compare their, you know, various, you know, as you know, I mean, even so-called fixed charge force fields, I mean, they're pre-polarized, okay? And the way that's been done and the way there's so many parameters under the hood that, you know, different labs are getting, you know, different results in detail. But the big picture is that aside from, you know, going to your specific question, if you do this extrapolation, if you use a fixed charge force field, hexane is 0 field, okay? But that's not the gas phase frequency. Here you can see, yeah, in a polarizable force field, we get this this value. By the way, there are multiple polarizable force fields out there, right? There's not just one, okay? And in fact, in this paper that's on the archive, we actually compare some of those, working with Richard Bradshaw in England. So you know, it's complicated, but here, yes, this extrapolates almost exactly. In fact, we could add that point onto this plot.
Yeah, I was wondering about that because I took this paper that you're talking about here not too long ago, and I just did this with acetone, and it did not hit the gas phase frequency.
Really? And you used a polarizable force field?
I just took your numbers from the paper to try it, which may or may not be valid, but I took that amoeba force field.
Oh, well, you have to do this. We've never.
Yeah.
We've never
Right.
done acetone. We did acetophenone for the reason you could see, because it can be incorporated into proteins as a non-canonical amino acid. But well,
You
that's
still would have
interesting.
expected. You still would have expected to get
I
the right
would.
value.
I would
Yeah.
have. So
Right.
so I urge you to look at this paper that we just uploaded a few days ago on penicillin and some modified versions of penicillin. And it turned. If I go back to that, I'm just to give you the big picture story. Here's penicillin. So this side chain conformation matters. And of course, this charge matters. And force fields have a hard time dealing with that charge.
Right.
The side chain we can deal with because you have different conformations, and you can do an ensemble, of those conformations and essentially correct for it. But if you really want sort of what's the essential core of this, you have to make this molecule, which is called PenM. And when we do that, that's a rigid molecule. It behaves exactly like acetophenone. And so it behaves very well. Whereas penicillin itself, you would get completely confused we got completely confused by looking at the solvatic chromosome. And that paper goes through every conceivable thing we could do, but we're really leaving it then to, people who are better at this than we are to give us a better approach. We've used everything that's out there, and we're working with, Bradshaw, and he's a state-of-the-art guy. So
thank you.
Yeah, I mean, I just to say something that people in my lab hear often, it's amazing that in 2021 we would be worrying about something so simple as the interaction between the solvent and the solute.
I know.
And the interesting thing, and it really is an interesting sort of thing to think about, and the person in my lab is thinking very much about this, is that people have thought about this for electronic transitions for a long time, solvatic chromosome. And the problem there is, where is the observable, to put it that way. Delta mu is somewhere, but the molecule's big, okay? And it has all this functionality on it, and that functionality is certainly going to be affecting things. So a huge amount of work has been done on that. The nice thing about the vibrations is that they're local, okay? But then the question is, how does this local thing and the more global thing, how do they relate to each other? And we don't know the answer to that yet. In fact, again, I keep referring back to Marilyn because I expect she's read every paper we've ever written. Many years ago, in fact, what got us started down this path was the observation that a nominally inversion symmetric carotenoid bound in LH2, the antenna complex in bacterial photosynthetic organisms, has a huge electronic Stark effect, huge delta mu. Now, how can something which, if you take it out of the protein, it's very polarizable, but delta mu is small. And so our assumption at the time, it's still one of the largest delta mu's ever measured. Our assertion at the time was that the organized environment in the protein is creating a field, and that field's interacting with this polarizable chromophore, inducing a dipole, and that's what we're observing. But the carotenoid, spans the entire protein. So, what is the field? It's, it's not local. It's some global aspect. I'd love to see somebody take on that problem, because it's really a big effect, and it's what then drove us to thinking that the fields from proteins could be big. And then you realize you encounter that in the retinal literature already. It was already described. Rich Mathis had done some work on this a long time ago. And it led us to look at vibrations, because there you can look locally. Now, what would be the ideal experiment? Well, here it would be ideally you look at, each one of these vibrations in the, separately. Unfortunately, not all of these modes are local. We're doing exactly this experiment right now, where we're taking a chromophore where we have electronic Stark measurements, and where we're positioning probes around the molecule so that we can sort of map the fields locally and then try to connect global and local. It's a very challenging problem, which may or may not be interesting. I think it's interesting. I'm not sure anybody else will.
Okay. I do not see any other questions arriving. So I would like to thank again our speaker of today, Stephen. Thank you very much for your very interesting presentation and
well, thanks for coming. And thanks for staying.